Chapter 10, this deals with controlling the microbial growth in the body, specifically looking at antimicrobial drugs. So previously we've talked about how do we control microbial growth in the environment. Now we're specifically looking at the body. Over years, from a historical standpoint, we've often used various drugs as a means of trying to affect the physiology in a detrimental way of the microorganisms. Chemotherapeutic agents will act against diseases. Antimicrobial agents or antimicrobials have been used to treat infections. And as I said, there's a historical standpoint of this. Paul Allergic used what was referred to as magic bullets at the time to kill microorganisms. They were actually arsenic compounds, so those are not used today. Alexander Fleming is known for discovering penicillin, which is an antibiotic that is released from penicillium, which is a fungus. And then there have been additional people throughout history who have discovered different additional drugs that have antimicrobial properties to them. And then Salman Waxman is the one that's credited with these antibiotics. These are technically, an antibiotic is a chemical that has antimicrobial properties to it that's being produced naturally by other organisms. So penicillin is an example that it's produced by a fungus, but it has antimicrobial properties against certain bacteria. This plate is showing with a particular species of penicillium. As I said, it's a fungus, and it produces penicillin, which is the antibiotic. And on the petri dish, what happens is the penicillin, the chemical, is released into the media and diffuses away, it moves away from the fungus. And that is why there's an area right next to it where you see absolutely no growth of, in this particular case, the bacterium that was used to inoculate the plate was Staphylococcus aureus. So you can see how it does inhibit Staph aureus from growing. Semisynthetics are antibiotics that have been chemically altered. Usually it makes them a bit more effective than the natural wild type. They tend to last longer, and they might be a little bit easier to administer than the original natural occurring ones. And then synthetics are antimicrobials that are completely made from scratch in the lab. Different mechanisms of antimicrobial action. We're going to look at this. One thing to keep in mind is that any successful type of chemotherapy, using medication, using drugs to help treat certain diseases, it requires selective toxicity. In other words, you want it to kill the microorganism, but not your cells. Antibacterial drugs are the largest number of any type of antimicrobial agents. Usually the drugs are effective against bacteria. That's what we're saying. These are the largest group of them. There are fewer drugs to treat eukaryotic infections, such as fungal infections, and even fewer to treat viruses. And we'll talk about why that's so. Some common sources of antibiotics and semisynthetics is listed here. As you see, some of the organisms are fungi and others are bacteria. And you can see in the right column the antibiotic that they naturally produce. And you might be thinking, well, why say is a bacteria producing an antibiotic in the first place? Or why is penicillium producing penicillin? Why is it doing that? It gives it an advantage. I mean, there's competition at the microbial level for resources, just like you see competition in general biology for food resources, water resources, etc. And so if you can inhibit anything from growing near you, that means all the nutrients near you are for you. Basically, think of a toddler that's never been told or taught to share. What are the mechanisms of how these various antibiotics work, or from antibiotics to the synthetic ones? Anything's antimicrobial. How does it work? There's 6 main categories of how they can work. Inhibition of the cell wall synthesis, inhibition of protein synthesis, disrupting the cytoplasmic membrane, inhibition of just general metabolic properties, pathways in general, inhibition of DNA or RNA synthesis, and inhibition of the pathogen's ability to attach to the host cell or to enter into the host cell. One thing that we look at in terms of effects of antimicrobials on cell wall synthesis, for bacteria, this becomes real important in noting that bacterial cell walls contain that peptidoglycan. They are the only organism that does produce the peptidoglycan. And so if you can target that, that's an example of selective toxicity. If you target interference with production of the building components of peptidoglycan, the NAG and the NAM, then that is very specific for the cell wall bacteria. Nobody else has it, so it's not going to affect any other cells. So I said most of the antibiotics that do interfere with cell wall synthesis will interfere with the peptidoglycan component of it. Some of the cross linkages that may interfere. Beta-lactams are a very huge group. They form what are known as the beta-lactam rings, and then they bind enzymes that will cross-link. But basically what's going to happen is the cell wall is going to be weakened, and eventually it's going to lyse. And this is showing the inhibitory effects of those beta-lactams in it. Semi-synthetic derivatives of beta-lactams tend to be a little bit more stable than the wild type or natural type. They're more readily absorbed. They're less susceptible to deactivation. And they tend to be effective against more numerous types of bacteria versus the naturally existing one. So that's an advantage of, if you're wondering, why don't you just use the natural one? Why did you start with the natural and then make it modifications to it to make it the semi-synthetic? These are reasons why. Some examples of antibiotics that are effective against bacteria that interfere with the cell wall synthesis would be vancomycin. Cycloserine is another example. A lot of those are more effective against your gram-positive bacteria. Bacitracin also is one that interferes with the cell wall. So this is giving a list of some of your more common antibiotics and how they do work. Sometimes it may prevent bacteria from increasing the amount of peptidoglycan. So if there's any injury site, there's no way to repair it. The existing peptidoglycan layer is fine. It's just you can't repair any. The other thing here is for any cell that is growing, it's going to interfere with that. Inhibition of the synthesis of fungal cell walls. Fungal cells do have a cell wall. It is not made out of peptidoglycan, so it's going to be different than bacteria. And animal cells do not have a cell wall at all. So it is possible to find antimicrobials drugs that are effective against interference with the cell wall synthesis of fungi. They can interfere with the glucan, which is one of the building components of the cell wall in fungi. But just keep in mind, if you have an antibiotic that's effective against inhibiting the cell wall synthesis, if it's effective against bacteria, it will not be effective against fungi because their cell walls are chemically, they are very different. Inhibition of protein synthesis. Something to keep in mind is that prokaryotes, when they are carrying out protein synthesis, the ribosomes, which remember that's a site for protein synthesis, are a 70S. You have two subunits, a 30S and a 50S subunit that combined form the 70S ribosome. Eukaryotes also have two subunits, but they are 40S and 60S. They're slightly larger, and they form an 80S. Essentially, they're not the same. So drugs can selectively, number one, target translation, the production of protein, and can distinguish between the 70S and the 80S. Now, the catch that you have to be careful of, if you develop an antibiotic that is effective against the 70S ribosomes, the thing you have to keep in mind is that, yes, that's found in prokaryotes. But if you remember back whenever you've studied about the cell, is that in humans, in our mitochondria, in the inner matrix, there are some 70S ribosomes. So over long term, it could cause some adverse effects by interfering with those 70S ribosomes and the protein synthesis that occurs there, which is occurring inside the mitochondria. And this is just showing the process of translation and step-by-step on how you can target those prokaryotic ribosomes to inhibit the protein synthesis. You can inhibit, and there's lots of different sites where you can inhibit, so cause the mRNA to be misread. You can cause problems with the large ribosome from coming in. Basically, then your tRNAs cannot bind in. They can interfere with the peptide bond formation that's supposed to occur between the two amino acids that are being held in site. And so there's multiple steps along the way that you can interfere with the protein synthesis. Some drugs will disrupt this cytoplasmic membrane, that cell membrane. They damage the integrity of it. Depending on which drug you're using, sometimes, yes, you have to be careful of, could there be potential side effects, because the basic structure of the cell membrane is very similar between animal cells, which would be us, and say fungal cells and bacterial cells. And so sometimes drugs can interfere. Basically, it's like making a pore or an opening, and so the inside of the cells can leak out. Some of them will interfere with the steroids that are present to help stabilize the integrity of the membrane. They interfere with that. Some antibiotics will interfere, like polymyxin, with the membrane of gram-negative bacteria. Problem with that is, over long term, it can become toxic to human kidneys. Some antibiotics are going to interfere with the various metabolic pathways, the various metabolic reactions that are occurring. They may interfere. Maybe they interfere with the electron transport chain. Maybe they inactivate some of the enzymes along the way so that the final product cannot be produced. So there's numerous ways that this can work. It depends on which drug you're looking at. Some drugs will interfere with the activation of viruses, so they can never replicate. Once again, this is showing some examples of whether it's interfering with folic acid synthesis. Some of them will interfere, like I say, with viruses, like in the uncoating and the reproduction of it. And then other ones can interfere with the nucleic acid synthesis, the synthesis of DNA for replication or RNA during transcription. The problem here is that because all cells, whether they're eukaryotic or prokaryotic, have DNA, have RNA, the potential for side effects greatly increases. So usually this is not used to treat infections. They are looking at it as a way of trying to potentially use it for treating cancer because cancer cells are going through replication much faster than normal. And so the idea is, well, if you could block that DNA replication, the cell doesn't go through division and it stops the cancer. The trick is trying to figure out how to do this without interfering with your normal cells. Sometimes there are analogs for the nucleotides. That's going to disrupt the shape. You disrupt the shape, you disrupt the function. And this is showing some of the various nucleosides and their analogs. Some of the various drugs that have been used against nucleic acid synthesis or like DNA replication, and inhibiting that might interfere with the DNA gyrase that has to help to unwind the DNA before replication takes place. Some of them will inhibit RNA polymerase, so transcription will never occur. Reverse transcriptase inhibitors. All right, humans do not make reverse transcriptase. That is an enzyme that uses RNA as a template to make DNA. So you might think, okay, well, why would you have an inhibitor if we don't even have that? Well, it's an example of that is the HIV virus does produce a reverse transcriptase. This is the way that virus will replicate. So if you can inhibit that enzyme, then that means the virus cannot replicate at all. Is it going to have a harmful or adverse effect on us as humans? No, because we don't have that enzyme in the first place. If you don't have it, it can't do anything harmful to you. Oftentimes, drugs are looked at in terms for preventing viral infections is to try to prevent the virus from ever getting into the host cell, your cell, to begin with. It's harder to treat something when it's inside your cell without harming your cell. That's very tough. So oftentimes with viruses, the idea is, okay, well, let's attack it before it ever gets inside your cell, because that would be a lot easier. So the virus has to attach to your cell, has to attach to the host cell. So some drugs are looking at preventing that attachment from ever occurring. If it can't attach, it can never penetrate. Some look at preventing once the virus attaches to a host cell, it still has to get into, it has to penetrate, it has to enter. And so preventing that step. And then once a virus is inside of you, it's a virus, the DNA or the RNA, depending on which it is, has a coat around it, protecting it. And so we refer to it as uncoating once it's inside of you. So if you can't prevent it from attaching and you can't prevent it from penetrating, then maybe you can prevent the uncoating. So essentially, it's looking at all the stages for viruses of their life cycle and how they replicate. And there's various drugs that can interfere with the various steps along the way. It's kind of like, well, if I can't stop you at point A, then I'm going to develop a drug that can stop you at point B. Well, point B doesn't work, then we got something for point C. So what is your ideal antimicrobial agent going to theoretically look like? Well, and if you're trying to put this in perspective, as I am making this particular video, we are in the middle of the COVID-19 pandemic. So in terms of trying to come up with something that would be effective at treating COVID-19, what are you wanting to look at? Well, you want something that's readily available. The number of cases worldwide for COVID-19 that we have experienced, obviously, it's a pandemic. It is not just a few people in a very isolated area. You want something that you can use to treat everyone, whether they're in the United States, whether they're in Africa, whether they're in Europe. It doesn't matter. You want something that's readily available. Ideally, you want something that's inexpensive. It has to be not only readily available for everyone, but everyone needs to be able to afford it. It needs to be chemically stable. It needs to be very easily administered and obviously non-toxic. You don't want people to form allergies against it. Now, ideally, when you're trying to develop an antimicrobial agent, it's nice if it can be effective against a wide range of pathogens. Why? Because it's easier if you can just use one drug to treat several different things. At this point in time, if we could find a drug that would be effective against COVID-19, even if it's very selective for this one particular virus, everyone would be absolutely thrilled. Now, reality and theory are two different things. In theory, it would be wonderful if we could find a drug that's already here that's effective. Because it's a virus, that limits things. A number of different pathogens that the drug acts against will determine how we classify it. Narrow spectrum is effective only against a few organisms. Broad spectrum is effective against many different organisms. There's pros and cons of this. When you have an infection, do you want to wipe out everything that is inside you? No, you don't. So from that standpoint, a narrow spectrum, it's like you just want to kill what's causing the disease. You don't want to kill off the good ones. However, do you necessarily want to have, I mean, think of, say, a pharmacist. How many drugs do they have to keep in the pharmacy? If everything was a narrow spectrum, you're going to have to, okay, I need drug A for organism A, drug B for organism B. I mean, it very quickly could just escalate to high numbers. So a broad spectrum is nice. Also from the standpoint, if you don't know yet what your patient has, you can administer a broad spectrum and it will hopefully kill what is causing the infection or disease, especially until you can identify it and then possibly switch to a narrow spectrum. The problem with the broad spectrum is that it is going to kill those naturally normal, good microorganisms that exist on us and in us. And then that can allow for secondary infections. It's like you're wiping the slate clean and now you've got all this extra space. Do you really want to allow that for another pathogen to come in and set up a secondary infection? No, you don't. This is showing you some of the different antibiotics, some of the more popular ones, and what they are effective against in terms of prokaryotes versus eukaryotes. And notice that there are more that are effective against the prokaryotes than the eukaryotes and viruses. So how do you measure the effectiveness? Well, you can run several different types of tests. One thing that you can do is what's known as a diffusion susceptibility test. It's often known as a Kirby-Bayer test. You grow bacteria on a lawn, basically just solid growth. You don't want to have individual colonies. So you just want that solid growth on there. What you do with these Petri dishes is, excuse me, you inoculate it with bacteria. And like I say, you just do this large, oftentimes you use a cotton swab and you dip it into like a liquid broth. So you just get that cotton swab soaked and just really swab it back and forth across that plate. So you get that good growth. At the same time that you inoculate it, then you put, each one of these white discs is representative of a different antibiotic. And so if we look at the ones that we can see on here is, I believe TE is tetracycline. On the jar, they'll give you the name and the numbers representing the concentration of it. So that's going to be on your original container. You place those on the plate and then you incubate it just like you normally would. After 24 hours, typically 24 hours, you come back and you look. Notice those areas around some of the antibiotic disc, and you have that clearing. That's known as your zone of inhibition. So on the one that you can see that they have the white circle around, the S10, and the white dotted line around, that is the zone of inhibition. Between that line and the disc, there's absolutely no growth. Why? Because the antibiotic that was on that disc diffused out into the medium, and this particular bacteria is susceptible, so it prevented the growth of it. So you would say, okay, that particular antibiotic that's on that disc at that concentration is effective. It prevented the growth. It inhibited the growth. So that would be a good antibiotic. Now, if you want to start comparing some, if you look at this plate over on the left upper side, the TE30, there's no inhibition. The bacteria is grown right up to it. Obviously, that didn't do anything. So don't use that antibiotic for this bacteria. It's not going to help. If you want to look at the effectiveness, compare the zones of inhibition. You would actually take a ruler and you would measure from the edge of the disc out to the edge of that zone of inhibition. And so you can visually look at, but you could actually calculate the very top center one. It looks like it's ENO, looks like a 5 on there. That is definitely much better than the S10. Why? How do I know that? Because the zone of inhibition is larger. It prevented the growth of the bacteria to a greater degree. So you can compare, number one, what inhibited growth, what did not. If it did inhibit growth, compare those zone of inhibition. How large is it? The larger the zone, the more effective it is. You can also measure the minimum inhibitory concentration. And so what this says, you have the same antibiotic in here, but you're increasing the concentration of the drug as you move from left to right. And you'll notice that those little wells, which you've inoculated, they have the antibiotic in them. And you're measuring to see, remember, turbidity means bacteria is growing. So in the turbid wells, the antibiotic was not effective at that concentration. So this is where you can see, okay, well, maybe at a low concentration it's not effective, but if I increase it, then it's going to work. You don't want to give a greatly increased concentration of antibiotic if a smaller amount would be effective. So that would be one reason why you're trying to see what concentration will inhibit the growth. This is an E-test. It kind of combines the two previous tests. On the strip, you have the antibiotic at increasing increments. And so once again, you would inoculate the plate, you lay the strip down in the middle. You can see the zone is higher at the top. That's because the concentration, 256, is much higher than, say, 1. So you can go down and look at this, and it looks to be about 0.75 where the inhibition begins. So you know that if you have a concentration less than that, it's not going to be effective. But once again, you could give a concentration of 1. Do you need to give a concentration of 64? Why would you go with 64 if 1 is sufficient? Minimal bacterial concentration test, or an MBC test. Once again, you're trying to determine the concentration that of the antimicrobial drug that you need that will inhibit the growth. And so once again, you've got in tubes, you have the various concentrations you played out, and you see at what point do you have 0 colonies. So at the 8 micrograms per mil, you still had growth, but at 16, there were no colonies. So 16 micrograms per mil would be effective. Why do the 25? Well, until you do this test, you don't know what concentration. So that's all they're trying to do, is figure out at what concentration will be effective. Routes of administration. There's different ways that you can administer drugs, and some are more effective than others depending on the type of drug. Topical application is for external infections. Oral route, that's good because you don't need any needles and a patient can self-administer it. So when you're talking about ease of application of the drug, these are some things to keep in mind. Intramuscular administration is when you have to inject into a muscle. Intravenous is directly into the bloodstream. So those two are going to be a little bit trickier if it has to be applied that way, because not everyone can do that. Do you have to teach a patient how to do it, or does a health professional have to give the injection? A lot of people don't like needles, so that could potentially be a problem. You have to have a supply of needles if it has to be given either IM or IV. So you have to look at all of these things and consider all of these things. You have to know how you're going to distribute that antimicrobial agent to the infected tissue. Does it need to go in the bloodstream and to be carried to the infected tissue in that manner? And so you have to look at different ways of, if you need to get it into the blood level, into the bloodstream, how long does it take for it to actually get into the blood, depending on how you're administering it? Obviously, if you do IV, it's going to be there immediate. If you take it orally, it's going to take a while for it to even show up in the bloodstream, and the concentration, it's almost like it gets diluted out along the way, and so it's not going to be as high. Where do you need to get it? You need to consider several different potential side effects when looking at antimicrobial drugs and the safety of it. So toxicity, you have to look at, are there any adverse side effects? Potentially, the drug's going to be toxic to the kidneys, to the liver, to nerves, and sometimes you have to keep in mind short-term versus long-term toxicity levels. You need to take into special consideration when looking at toxicity of medications when dealing with pregnant women. Can it cross the placental barrier? And then can it cause adverse side effects to the developing fetus? The therapeutic index is a ratio of the dose of the drug that can be tolerated to the drug's effective dose. Some side effects, and keep in mind they're going to be showing you some of the most severe for kind of dramatic effects on the pictures in your book. But these are showing some of the side effects. Can turn your tongue black, turn your teeth yellow. Some people may develop allergies, and some of them may be mild. Some could lead to anaphylactic shock, which can be fatal. So you have to be careful of that. If you are given antimicrobial drugs to a patient, are you going to end up with a secondary infection or not? This is often a huge concern for patients in the hospital. Their immune system is already compromised. They're already sick. They're already fighting something, because nobody goes to the hospital for vacation. So they're there because they're already sick. It's going to put them at a higher risk of developing a secondary infection. If you were exposed to another pathogenic organism, if you are a healthy individual, it may be what we call an opportunistic one, and you're healthy, you're able to fight it off, you never know you were exposed to it. But for somebody whose immune system is already compromised, they're going to be more susceptible to it. And if you're taking an antibiotic where you just wiped out a lot of the normal organisms in you, then you just really made it a much better chance for a secondary infection. Some pathogens are naturally resistant. Other organisms are not. They're susceptible. How do they suddenly become resistant? Well, maybe there's a mutation that occurred. And some of them, what we have found is that on the plasmids is typically where we will find the genes for antibiotic resistance. And plasmids will replicate on their own schedule and can be transferred from one organism to another. How? By transformation, transduction, or conjugation. So you can have an organism that was susceptible, say, to an antibiotic, and it obtains the plasmid that has a resistance gene for it, and now it's resistant. This is just showing a diagram of how you have on the far plate on the left your microorganism, in this case your bacteria that are growing, you do have a couple of mutations in there, the red ones that are resistant to the drug. The organism are exposed to the drug. Well, all those that were susceptible died out. The two that were initially resistant because of mutations, they're going to grow. They're going to go through cell division and produce more, and go through cell division and produce two more, and continue on. And so now you've altered that population to now being more resistant. One thing that I often say is that drug resistance, antibiotic resistance, has really become a problem. Many years ago, microbiologists will tell you that it really has developed because of the misuse of and abuse of antibiotics. Years ago, it used to be fairly common. Somebody was sick, and the healthcare professionals are like, oh, let me just write you a prescription for an antibiotic, and this will help you. So part of it stems from the side of the patient, but part of it is the fault of the healthcare professionals, where they were too quick to prescribe an antibiotic. Sometimes wouldn't even run a test to see what you have. Oh, let me just write this prescription for this drug. And that happened a lot. And so a lot of people were taking antibiotics maybe when they really didn't need to. So what happens there? Well, you have a lot more increased exposure, which is going to increase the chance of resistant organisms. The other problem that has risen or played a role in this resistance to antimicrobial drugs falls on the patient. Years ago, most antibiotics, it was a 10-day regimen, 3 times a day for 10 days. And what would happen is the patient would come to the doctor, I'm really sick, the doctor would prescribe an antibiotic, and the patient would start taking it. Well, about day 3 or 4, it's kind of like, I'm feeling a little bit better now. By about day 5, you're like, I'm feeling really good. I don't need to take this medicine for a full 10 days. That doctor's crazy. What does he know? I feel great. So I'm not taking this medicine anymore. And so people would stop taking it. Have you killed off all the pathogens? No, you have not. You killed off most, but as long as there is one that is still there, guess what happens when you stop taking the antibiotic? The bacteria start growing. They continue growing, and now their numbers are going up because you're no longer taking the antibiotic to kill them. And so now about day 8, you're like, I'm not feeling so good anymore. And about day 9, you're like, that doctor, he just doesn't know what he's talking about because now I'm sick again. Now I'm going to have to go back to him. When in reality, if you had continued taking the antibiotics for the full regimen, it would have killed him. So the fault kind of lies in both places. Once we had started realizing that we were dealing with this huge problem as it's continued to get worse and worse, now that certainly the healthcare field has gotten much better about prescribing antibiotics only when necessary. And then really trying to educate the patients into the importance of you need to finish the whole regimen. If it's 5 days, you take it for 5 days. If it's 10 days, you take it for 10 days. You finish them until they are gone. Now, the other problem is with, like I said, where doctors used to just prescribe, oh, I've got a cold, here you go, here's some antibiotics. Something to keep in mind, antibiotics for the most part will not work against viruses. They're not going to do diddly squat for it. So, for most people, if you have a viral infection, they are not going to prescribe an antibiotic because it's not going to work. The only time that they will, and there are certain circumstances where it's okay to, you may have an individual whose immune system is compromised. Maybe they're going through cancer treatment. They already have enough going on. You don't want them to get a secondary infection on top of the viral infection. And so in that case, you may give an antibiotic to prevent a secondary infection from occurring. But for most people, you're not going to, like I say, with a viral infection, prescribe antibiotics. What's the mechanism of resistance? Well, there's different ways, and this is where what we're talking about now is with the microorganisms, and it's kind of like this. Sometimes I describe it like a chess game where you're going back and forth, or kind of like a war game back and forth. So we have these antibiotics, whether they are natural antibiotics or whether they are fully synthetic antimicrobial drugs, whose purpose, whichever one they are, is to essentially kill the microorganism, prevent the infection from getting worse, prevent the disease. It's to kill them. Well, in microbiology, we're always looking at two sides of everything, because we look at it from the side of us as humans. Yes, we want to kill these pathogens. But then we also look at it from the side of the microorganisms. So the mechanisms of resistance, this is looking at now from, say, the bacteria side. Okay, you have these drugs. How have I adapted so that you can't kill me? You want to kill me, but I got some tricks up my sleeve as to how I'm going to deal with this. We know of at least 7 different mechanisms. Number one, the microorganism may produce an enzyme that destroys the antimicrobial drug, whether it's an antibiotic. And this is true, like I say, whether it's an antibiotic, whether it's a semi-synthetic, or whether it's a fully synthetic antimicrobial drug. Whatever that drug is, the microorganism might deactivate it. It might slow or even prevent the entry of the drug into the cell. And if it can't get in the cell, it's not going to do anything to it. It may alter the target of the drug so it can't bind effectively. The microorganism might change its own chemistry, its own metabolic chemistry and reactions that it's doing. It may have the antimicrobial drug may be entering into the cell. Well, say the bacteria, that might be pumping it out as fast as it's coming in. It's kind of like with, I don't know, kids in the summertime. You're running in my front door, open the back door, out you go. In the front, out the back. In the front, out the back. Some bacteria in biofilms, it's kind of safety in numbers, where you have this whole community of bacteria, different species growing together, kind of gives extra protection. They can resist these antimicrobial drugs better than if they were individually by themselves. And then some of the bacteria, such as Mycobacterium, can produce certain proteins that will actually prevent the drugs the way they're supposed to bind, such as it's supposed to bind to the DNA gyrase. Well, they produce a protein that prevents the binding of the drug, so it's never going to work against them. Beta-lactamase, it's a penicillinase that renders penicillin inactive. And so this is an example of, okay, penicillin is supposed to kill me, but you know what? If I produce penicillinase, it's going to inactivate that particular drug. Now, not all pathogens are going to have all 7 of those, but they may have more than one type of method. They also may acquire resistance to more than one drug. And this is a problem that we have when we have multiple drug-resistant pathogens. That's when they are resistant to at least 3 antimicrobial agents. It starts to become a problem of what's left that you can use to treat the patient that's not going to have stronger side effects for the patient. Because typically your first line of choice for antibiotics are going to be those with the low toxicity, the low side effects for the patient. And so when your pathogen is resistant to that and you have to go to the next drug and then the next drug and then the next drug, you tend to be going to the harsher and harsher drugs. How do you try to prevent this resistance from occurring? Try to maintain high concentration of the drug in the patient for a sufficient amount of time. That inhibits the pathogen, basically trying to allow the immune system to work, to get rid of it. And sometimes you're going to use multiple drugs in a combination. And this is shown here where you've got the 2 discs. And you can see the zones of inhibition, but you can see where the two diffuse together in the center. They're working together in a synergistic fashion, and so it's much better. So use both drugs together. Once again, use antimicrobial drugs only when necessary. So like I said, antibiotics against viruses, there's very few because viruses, the reason there's fewer drugs for treating viruses is that the virus is going to spend most of its lifetime in your cell. The only time it's not inside your cell is when it's been released from one of your cells and it's traveling to the next cell. It's a very small time period, very tiny window. So the question comes up, how do you kill something that is inside your cell? How do you even know it's in there? How do you kill it without damaging your cell? That gets very, very tricky. And why are there fewer antimicrobial drugs against fungi as well? It's because fungi are eukaryotic cells. Their cells, animal cells are eukaryotic cells. So fungal cells are much more similar to animal cells than, say, bacteria. Yes, fungi have a cell wall, so we can try to use that as something to work against. But the ribosomes are the same as ours. A lot of their metabolic processes are the same as ours. And so that gets a little tricky once again. How do you kill it without killing your own cells? How do you, it's best to find the differences. Well, how do you find the differences when there's so many similarities? And that's where it gets tricky. We're constantly doing research to develop new drugs and make variations of existing drugs. So that's what we're talking about. Second generation, third generation is where you have an existing drug and you make some modifications to that. And then, as I mentioned before, we're constantly looking for new antibiotics. We're trying to stay one step ahead, essentially, of the microorganisms. They develop ways to counteract the ones that we've come up with, so we're trying to stay another step ahead. And once again, the easiest thing is try to look for those differences.